Hello students, once again welcome back to my channel. Today we are going to start the next topic in sexual reproduction in flowering plants. That is a structure of Microsporangium or structure of a mature anther. Okay, so in the previous lecture we have discussed about the structure of a stamen. What are the different parts of the stamens? The detailed aspect of a stamen we have discussed in the previous video. So we will move towards the structure of a microsporangium. So microsporangium is otherwise also called as anther. See, when we take the transverse section of stamen, so when we take the transverse section of a stamen or anther, so anther it is the terminal part of the stamen, and this is the anther. When we take the section of transverse section of anther, it reveals the following feature. Since we know that a typical microsporangium structure, when we see under the microscope, it reveals this structure. So this is the outline of the microsporangium. Outline of the, it is almost circular in its structure, and this is the diagram showing the enlarged view of the microsporangium. So first we shall see this structure. So circular in this structure, what are the different layers of the microsporangium? Let us see. Number one, the epidermis, the outermost layer. It is followed by endothecium and middle layer and tapetum layer, and the innermost, the central cavity of the microsporangium or the central part of the pollen sac, it is also called as, it is filled with microspores. So this is the structure showing this is the outline of the tiers of microsporangium or end anther. Okay, so when the structure is enlarged and see under the microscope, so what are the structural features it is revealing? Let us understand. Its function also we are going to discuss. So first of all, epidermis. Epidermis is the outermost layer, which is single layer made up of compactly arranged cells, barrel-shaped cells, and it is a protective in its function. It protects the inner tissue. It is followed by endothecium. As you can see, endothecium is also a single layer. It is also made up of compactly arranged cells. And what is the role of the endothecium? Here, endothecium is not only protects the inner tissue, it also play a very important role in the dehiscence of the anther. So as you know students, when the anther get mature fully, at the maturity, the dehiscence of the anther taking place and releases large number of microscope outside the microsporangium. So that is a function of the endothecium, and it is playing role in the dehiscence or splitting of the anther. Let us see in the next layers, middle layer. As you can see, 2 to 3 layers, 2 to 3 layers or sometimes 3 to 4 layers of endothecium, middle layer layers are there. Then again question arises, what is the role of the middle layer and it is made up of what type of cells? You know that middle layer, or it is 2 to 3 layer, it is usually made up of parenchymatous cells. As you know the function of parenchyma cells, they store the reserve food material, and here also they are made up of parenchyma cells only and store abundant amount of food material, that is middle layer. Okay, so middle layer not only helps in the storage of food material, and it also protects the inner layer, inner tissue also protected by middle layer. Thus the entire structure of when we see under the microscope, epidermis, endothecium, middle layer, these three are not only protecting the inner tissue, and they are playing the different role during the dehiscence also, and they store the food material also. Let us move towards the next innermost layer. The innermost layer of the anther, young anther, what it is made up of? It is made up of a single layer of a layer called as tapetum. Tapetum is once again a single layer, compactly arranged layer, and it is having dense cytoplasm. It is having dense cytoplasm and contains a nucleus. Sometimes the nucleus may be one nucleus or sometimes more than two nuclei. It may be mononucleate or it may be binucleate, and sometimes multinucleate also. So do you know students why they become binucleate or multinucleate? So there is one reason. That reason is endomitosis. So during the division of the tapetal cell, here tapetal cells along with the nucleus also they divide. So due to endomitosis, this is the one reason why they become binucleate or multinucleate. Means the only reason for this is that is endomitosis. This is a key word. You must remember the key word. This word is very important. So due to endomitosis, the number of nuclei is going to increase in their number, having abundant amount of cytoplasm within the tapetum layer. And what is the role of the tapetum layer? So the main role of the tapetum layer is to nourish the developing sporogenous tissue. Then what are sporogenous tissue? So nourishment is very important. So tapetum layer take the nutrition from the middle layer and supply the nutrition to the developing microspores. In future, these cells called as microspore mother cells. Firstly, they are how they appear. They appear very compact. As you can see, there is no intercellular spaces. They are very compactly arranged. So the mass of sporogenous tissue, they are undergo mitotic division and they form large number of sporogenous tissue. And further, they undergo division, meiosis. They undergo meiosis and they produce large number of microspore mother cells. So here mitosis leads to later on it takes place meiosis. Sporogenous tissue undergo reductional division or meiosis to give rise to large number of microspore mother cells, and these microspore mother cells or pollen mother cells in future they produce large number of pollen grains. So this is how the structure of microsporangia shows its features. Okay. Okay. Apart from this, what is next? So here, this is the enlarged structure I have explained you. So after studying the enlarged structure of microsporangium, there is a small one concept is mentioned at the end of the, in the NCERT reader. After this concept, the small concept is given in our reader, and that is microsporogenesis. So apart from learning the structure of and TS of anther, we will also learn the next concept, that is microsporogenesis. Do you know, students, before going to study the microsporogenesis, we want to learn some more aspect about the TS of anther. One point I have to explain here, that here large number, here how many? They are tetrasporangiate structure it is. TS of anther is outline, it is circular, but having how many microsporangium? There are 4 microsporangia are there, or they are also called as pollen sacs. Within the pollen sacs, large number of pollen grains are present. Okay, and these are the different layers of the microsporangium. So each microsporangium composed of large number of microspores. Okay, and at the maturity, the dehiscence occur. So dehiscence occur and large number of microspores are released outside. And one more point I have to highlight here, that during the maturity, when the anther is younger, tapetum layer supply the nutrition. But when the anther get mature fully, dehiscence takes place, the tapetum layer disappears. Okay, so this is a complete structure of microsporangium. So next, the another definition, let me clarify now itself, that is the definition of a microsporogenesis. What is microsporogenesis? In one line I have to define it. Microsporogenesis is nothing but, so it is a process of formation of microspores from microspore mother cell through meiosis is known as microsporogenesis. So there is a very important definition, and diagrammatic representation also I will show in the next video, but definition you must learn. So large number of sporogenous tissue, they undergo meiosis. They undergo meiosis and give rise to large number of microspore mother cells. Sporogenous tissue undergo meiosis and give rise to microspore mother cells, and these microspore mother cells, their ploidy is haploid. They produce spore tetrad. Later they produce spore tetrad, and each tetrad consists of 4 microspores, and each one will get separate, and they produce large number of pollen grains or large number of microspores at the end of the microsporogenesis. So this is a complete concept, a structural aspect and definition of microsporogenesis we have discussed, and the rest of the thing we will discuss in the next video. I will stop here only.